Howdy everyone, my name is Sergio Lozano,
My name is Roberto Blanco.
and today we're going to be presenting on humoral immunity. So we first want to talk about the humoral response, the phases and types of humoral immune response. The humoral immune response is an essential component of the adaptive immune system that consists mainly of B cells and the antibodies they secrete to combat extracellular pathogens such as bacteria, viruses, and toxins. It becomes activated when B cells encounter antigens, usually in conjunction with helper T cells. The activated B cells proliferate and differentiate into plasma cells, which secrete antibodies, and memory B cells, which ensure long-term immunity. These antibodies work by neutralizing the pathogens, marking them for elimination, and triggering the complement system. There are two broad categories of humoral responses that consist of T-dependent, which require the helper T cells and lead to more robust, durable immunity and isotype switching, and T-independent, which is a response to non-protein antigens that and primarily involves the production of IgM with an earlier response and little or no memory. Antibodies also differ in function. IgM is the initial response, IgG is the long-term immunity, IgA is defense against mucosal surfaces, IgE repels parasites and triggers allergies, and IgD activates the B cell. So moving on to the stimulation of B lymphocytes by antigens. Activation of B lymphocytes starts as an antigen and it is bound to the B cell receptor on the surface of the B cell. This differs according to the nature of the antigen. T-dependent antigens, typically proteins, are taken up by the B cell, degraded and displayed on MHC class II molecules. The helper T cells recognize this complex and deliver the requisite signals in the form of cytokines and direct contact, and thereby completely activate the B cell. This gives rise to vigorous immune responses such as class switching, generation of high-affinity antibodies, and formation of memory cells. T-independent antigens, however, like polysaccharides or lipopolysaccharides, are able to activate B cells in the absence of T cell help. These usually cause a faster but weaker response with the predominant production of IgM antibodies and minimal or no immunological memory. Moving on to the roles of innate immune signals in B cell activation. Innate immune signals play key supporting roles for B cell activation. Pattern recognition receptors such as toll-like receptors and B cells and other immune cells detect PAMs or pathogen-associated molecular patterns during an infection. Stimulating these innate receptors either directly on B cells or indirectly on surrounding innate immune cells like dendritic cells provides additional activation signals that enhance B cell responses. These signals have the potential to increase antibody production, enhance class switching, and increase the overall immune response. Innate signals are also capable in certain instances to facilitate the activation of B cells that react with T-independent antigens and are therefore an essential bridge between the innate and the adaptive immune system. Moving on to heavy chain isotype switching. Class switching is a process by which an activated B cell switches the class antibody it produces from IgM to IgG, IgA, or IgE, but it's not antigenic specific. Switching is controlled by the T helper cells and cytokines. Each isotype has a specialized function. IgG is the most common in the blood and provides long-term protection. IgA is found in the mucosal areas. IgE protects against parasites. IgM is the default first antibody, and this switching allows the immune system to tailor its response to different types of threats.
Okay, let's go talk about affinity maturation. Is the generation of antibodies or ever greater affinity for the particular antigens by B cells. It takes place on the germinal centers of the lymph nodes following B cells activation through a process known as somatic hypermutation. Random mutations are introduced into an antibody variable gene. B cells that produce higher affinity antibodies are selected and permitted to survive and proliferate. This leads over time to generation of antibodies that bind more specifically and affirmly to the antigen, enhancing the accuracy of and strength of the immune response. Let's talk about the antibody feedback. Antibody feedback is a crucial mechanism that keeps our humoral immune response in check and stop it from going overboard. When antibody levels start to climb during the immune response, some of these antibodies latch onto the antigen, creating immune complexes. B cells that come across the complexes through the B cell receptors Fc receptors get the inhibitor signal. This signal essentially tells the B cells that there's already enough antibodies floating around, which helps deal down for the activation of antibodies production. This negative feedback loop is vital for maintaining a healthy immune balance and preventing unnecessary or excessive immune reactions that could harm or our healthy tissues. Okay, so let's talk about effector functions of antibodies. The antibodies help the immune system identify and eliminate harmful invaders like bacteria and viruses. Once antibodies bind to a target, let's call it an antigen, they trigger different effector functions that protect the body. That can be neutralization, that antibodies block viruses, toxins, or bacteria. Opsonization, antibodies stack invaders to make them easy for immune cells. Complement and activation, that is, antibodies activate the complement system, a group of proteins that help kill bacteria by pushing holes on their membranes. Antibody-dependent cellular cytotoxins, ADCC, antibodies that help natural killer cells recognize and kill infected or cancerous cells. Agglutination, that's antibodies cause pathogens to clump together, making it easy for the immune system to clear them. And then let's talk about vaccines. We have different types of vaccines, like attenuated vaccines. Weaken microbes that cannot cause disease but will still trigger a strong immune response. Subunit vaccine use parts of microbe to like proteins or sugars. Conjugated vaccine combines a sugar and proteins, so helper cells are stimulated and make a strong antibody response. Synthetic vaccine is lab-made proteins that look like a part of the microbe. Viral vector vaccine uses a harness virus that deliver microbial genes into the cells to make an antigen and trigger an immune response. And we have, by the end, DNA vaccines use the DNA to tell the cells that make a microbial antigen. And that will be it. Thank you so much.